The George U. Foundation of Nutrition and Health is proud to present Starving Cancer Cells to Death, Part 2. This is a tribute to Dr. Peter Peterson, professor at Johns Hopkins. We wanted to give you this as a special gift because of the seminal work and its implications today. It was published in 1978, yet it has all the important information that we all need. This is at no cost to you. It talks about cancer energy. It talks about the membrane deviation. It talks about calcium dysfunction. And it talks about the lack of mitochondria in cancer cells as a prognosis of how aggressive it is. Last year, I was given these two flasks, which came from the original lab of Dr. Otto Warburg, the winner of the Nobel Prize in 1931, the head of the Kaiser Wilhelm Institute, oddly enough funded by the Rockefeller Foundation. He was the only man of Jewish descent that was left and kept as a treasure in Germany during and before World War II. In 1978, when I was on my research year with Dr. Donald Coffey, I made two discoveries. Dr. Albert Leninger, head of the biochemistry department, was talking about cancer bioenergetics, meaning how they feed. And then I met Dr. Peter Peterson, who was the expert on cancer mitochondria. And these two men left a major imprint on the way I thought as a surgeon and as a doctor. Since 1964, Dr. Peter Peterson has been at Johns Hopkins and has led the lonely road as a hero of cancer metabolism, even when the genome discovery made it so that everybody switched from metabolism to molecular biology. These illustrations show how flabbergasted I was when I saw that the mitochondria became more and more abnormal with a more anaplastic type of tumor. It could easily be a prognostic tool for all of us to know how aggressive a cancer is. It's not necessarily in the nucleus. It's in the mitochondria, only seen on electron microscopy. Today we have tools, and just to give you an overview, the cell has these little energy packets called the mitochondria. And these mitochondria in cancer cells are clearly abnormal. And what he says is 50% less mitochondria in very aggressive cancers. They have an abnormal use of their fuel in that they use not only sugar, amino acids, oils, and even ketones. We think right now that most cancers use sugar, but not true. And you could see that the cancer cell, as opposed to a normal cell, uses 60% fermentation or glycolysis and only 40% oxidative phosphorylation, which is respiration, whereas a normal cell uses 10% fermentation and 90% respiration. Here's a typical PET scan showing a brain cancer, which uses heavy amounts of sugar as their main source of energy. And the FDG is but an analog of glucose showing up that we can see. It can be used both as a diagnostic and a prognostic tool of whether the therapy is working by the amount of sugar that these cancers take. 80 to 90% of cancers show FDG positives. Here's an ultrastructure done by Dr. Pete Peterson. On the left is a normal mitochondria, beautifully shaped, whereas as you go to the right, this is a very abnormal mitochondria. And this is what you see in aggressive cancers. Their source of energy is totally aberrant and invasive and aggressive. This is an ultrastructure slide done by Dr. Robert Elliott in New Orleans showing breast cancer. And you can see on the left slide a swollen mitochondria. and on the right, a totally abnormal mitochondria by the arrows. This is typical of every cancer. What Pete Peterson said was these abnormal cancers have less than 50% mitochondria, so it's not very efficient. It's not using oxygen well, if any. The more aggressive the cancers, the more abnormal looking. The more aggressive the cancer, the more it finds different sources of energy. The primary energies are sugars, as we all know, carbohydrates, but glutamine is also one, and other amino acids, and even fatty acids, and even ketones, as it evolves from a slow-growing cancer to a more aggressive one. In light of this, we need to understand today it is very popular for people to use ketogenic diets, and this may also have its flaws. The important points from the cancer mitochondria bioenergetics are the following. You can read this in detail as we will give you the whole document after this presentation. Number one, there is a degree of abnormality in mitochondrial dysfunction depending on how aggressive the cancer becomes. Mitochondria can use both glycolysis and an abnormal form of respiration using oxygen. Dr. Peterson discovered hexokinase 2 in mitochondria, which is especially characteristic of cancer cells. Aggressive cancer cells do not only use sugars, but can adapt to other sources such as pyruvate, glutamate, malate, galactose, ketone bodies, and acetoacetate on page 215. The more aggressive the cancer, the more deviant the energy source. Cancer mitochondria will become swollen as they become abnormal. Cancer mitochondria membrane have a unique feature such that they cannot get rid of calcium as well as normal cells. Cancer mitochondria membrane have a higher cholesterol to phospholipid ratio. This phospholipid ratio is also different from normal cells, and there may be future avenues of manipulating the membrane of mitochondria. The mitochondria DNA may become abnormal by an insult externally and then lead to total cellular and nuclear abnormality. Now, you can explore some of the work done by Dr. Pete Peterson in many of his sites, and this seminal paper that he wrote in 1978 has been cited 745 times. Therefore, I urge you all to read this at no charge to you. Combining the history of caloric restriction diets and cancer and the science of bioenergetics by people like Dr. Peterson, our foundation believes that limiting food and calories is the first form discovered of starving cancer cells to death using a metabolic approach to stop cancer growth. In 2002, I was the auditor to present at the NIH the calorie-restricted diet on terminal cancer cases. Out of 300 cases, we found that all these 6 cases presented showed shrinkage of tumor without any further treatment. Now, in this slide, I audited the Kushi Institute, the Hippocrates Institute, and others, including a visit to Okinawa, to look at 3 different types of diets, and I found one thing common. They all ate low-calorie, high-nutritious foods. So why is it that it works? What did I learn from these 13 years or more of monitoring these people's diets and watching the terminal disease change? They all ate low-calorie diets of 1,500 calories or so. The nutrition was very dense. They all lost 10% of their body weight, which is frightening for some people, but actually they felt great. On CAT scan, we noticed that they have what I call visceral defatting. In other words, their visceral fat shrinks at the same time that their tumor shrinks within 3 to 6 months. Their blood sugars all go into the range of 70 to 85 as fasting studies. Their insulin level goes down to 5. The hemoglobin A1C goes down to 5. The beta hydroxybutyrate ketones elevates in the urine and the blood. And what's interesting is their PET scans start taking up less of the FDG as they go on calorie restriction. The problem is long-term calorie restrictions presents problems like bone osteoporosis, sex hormones decline, and of course, thyroid hormone T3 decreases. One of the problems that I saw was 2/3 of these people did not do well at the end. About the third will do well, and even some are cured, which I cannot explain. So, when you see a person losing 10% of their weight, you're alarmed. And most of my colleague doctors are also alarmed. When they go on a calorie restriction diet, this is what happens. But what is interesting, comparing cachexia, which is the end stage of the metabolism of cancer patients, and what we call caloric restriction, and what Kotler quote as starvation, is that the insulin in starvation goes down, whereas in cachexia it goes sky high. Therefore, we're not surprised when we use calorie restriction initially that the weight loss is not a problem, and it is different from cachexia. So, Dr. Roy Wilford did the study of Biosphere 2 in the 1990s in Arizona, and he compared human evidence of calorie restriction to the animals. In the animal study above, you could see the monkeys of the same age, but one is eating a calorie-restricted diet, looking healthy, vital, and moving, and the other one on the right is old, looking, eating regular diets. Now, we could show that they retard age and cancers in worms, mice, rats, but we don't know about monkeys or humans. This was published in the New York Times in 2009. Looking at the human restricted study for 2 years in a very controlled environment of no toxins and self-grown food, you could see on this slide that looking at the left side of the slide, when they were in Biosphere 2, that they all lost body weight. All their blood sugars went down on the left, as you can see. On the right, they came out and they were finished with the study. The insulin level also tumbled, and the hemoglobin A1C went right down also. So this is the only human evidence of calorie-restricted diet in a 2-year period, and I'm sure there are more, but this is a classically very expensive study that probably will never be done again. In 2003, I had a moment of truth when I was operating at George Washington University Hospital for a long operating case as I came out for a break in the operating doctor's lounge, and I saw an article as I passed by on my way to the bathroom showing genomic profiling of short and long-term calorie restriction shows similar results. This was published by Stephen Spindler, and after the operation, I took this article home, and what he's saying is that short-term calorie restriction, meaning limited time, is almost as good as long-term, looking at 12,422 genes and gene expressions. In other words, even after a few days of lower calorie, your gene expression will start changing. By 2 weeks, half of your gene expressions will change to a younger person. And by 4 weeks or 1 month, 72% will change. This was really music to my ears because now I could give patients shorter stints of calorie restrictions for chronic diseases. Steven Spindler made a slide which shows that by using short-term calorie restriction, he can make all your gene expression into a more youthful, healthy expression. You can look this up on the website and Google him. Even very short calorie restriction like fasting has benefits on cancer treatments. Dr. Longo is also one of Dr. Roy Wilford's disciples, and he's modified this to show how it can be useful to all our healthcare and our patients. There are other metabolic approaches that are needed because not everybody does well, and I'm a great believer as a surgeon that we need multimodality to conquer this wide range of cancer diseases. There are these metabolic agents which block the cancer metabolic process, like dichloroacetate, palladium lipoic acid, and the most popular is the 3-bromopyruvate. These block the cancer metabolism without hurting the host cancer patient. The biggest article that was published was by Dr. Young Ko and Dr. Pete Peterson, and it describes their experience in 2004. At the present time, we're still investigating this molecule, and fortunately, with the help and facilitation of Dr. Leanne Jensen at the National Cancer Institute, in 2015, we took 60 human cell lines in a screening using 3-bromopyruvate, and at 100 micromolars, we could see significant death rate for leukemias, lung cancer, colon cancer, brain cancer, melanoma, ovarian cancer, renal cancer, prostate cancer, and breast cancer. This is only a screening. This is not in vivo studies, and this is in vitro. This does not interpret to real life experiences. So recently, Tom Seifert wrote a classic book called Cancer as a Metabolic Disease. And what his proposal is that the first insult of carcinomatosis may be a change in the mitochondria. Whether it's a mitochondrial DNA or mitochondrial membrane, we don't know, before nuclear DNA changes. This may explain why the study on Cancer Genomic Atlas Project failed, because that may be a secondary effect after the mitochondria changes. I urge you all to get this book, Cancer as a Metabolic Disease. You will never forget it, and it changed my life. We have asked Dr. Seifert to check and test and corroborate the findings at Hopkins of 3-bromopyruvate, and this is ongoing. Therefore, we need your contribution and your support. Tom Seifert wrote this in his book on page 235, which is very apropos. Respiration is responsible for maintaining genomic stability of a differentiated state like us. And respiratory insufficiency, meaning not having enough oxygen, will eventually induce a default state of unbridled proliferation or cancer. So, to prevent cancer growth and to promote health, we must always push for optimal respiration, remembering below that glycolysis does not use oxygen. It's anaerobic and only generates 2 ATPs of energy packets, whereas respiration will generate oxygen and lots more ATPs from 30 to 36 ATPs. And this is what we need to strive for to remain healthy. Again, please download this wonderful piece of work by Dr. Peter Peterson, and it's apropos today. We thank you for watching, and we hope you download this document and spread it to all your colleagues. Thank you and goodbye.